Bachelor of Science Nursing Microbiology, Unit 2, Culture and Media Preparation for Microbial Growth. Culturing microorganisms is essential for their identification, antibiotic susceptibility testing, vaccine production, and research. Microbes require specific nutrients, temperature, oxygen levels, and moisture to grow. Culture media can be solid or liquid and are classified based on their composition and function. Types of culture media. Culture media are classified based on their chemical composition and intended purpose. Based on composition, synthetic, chemically defined media. Exact chemical composition is known and controlled, used for research and microbial metabolism studies. Example: glucose salt broth supports minimal bacterial growth. Advantage: allows precise control over nutrients for bacterial growth. Used in studying specific nutrient requirements of bacteria. Semi-synthetic media contains both defined and complex components, natural extracts, used for culturing fastidious organisms. Example: blood agar contains agar and animal blood. Advantage: provides enriched nutrients for bacteria with complex needs. Used in isolation of pathogenic bacteria based on purpose and function. Enriched media contains extra growth factors, blood serum, vitamins, amino acids. Supports the growth of nutritionally demanding fastidious bacteria. Example: chocolate agar for Neisseria and Haemophilus species. Advantage: essential for isolating bacteria that do not grow on simple media. Used in diagnosis of respiratory tract infections. Enrichment media promotes the growth of specific bacteria while suppressing others. Example: selenite broth for isolating salmonella from feces. Advantage: enhances the number of target bacteria for easier detection. Used in detecting bacterial contamination in food and water. Selective media contains antibiotics, dyes, or chemicals that inhibit unwanted bacteria while allowing specific microbes to grow. Example: MacConkey Agar selects for gram-negative bacteria, inhibiting gram-positive bacteria. Advantage: helps in isolating bacteria from mixed cultures. Used in identifying foodborne pathogens like salmonella. Differential media distinguishes between bacteria based on colony appearance and metabolic activity. Example: eosin methylene blue, EMB, agar, E. coli appears metallic green due to lactose fermentation. Advantage: helps differentiate bacteria based on biochemical properties. Used in identifying enteric bacteria in clinical samples. Culture methods: solid and liquid media. Solid media contains agar 1.5 to 2%, which provides a firm surface for microbial growth. Used to isolate, differentiate, and identify bacterial colonies. Examples: nutrient agar, MacConkey agar, blood agar. Advantage: allows observation of individual bacterial colonies. Used in pure culture isolation in diagnostic labs. Liquid media, broths, does not contain agar, allowing bacteria to grow freely in suspension. Used for growing large quantities of bacteria or anaerobic bacteria. Examples: nutrient broth, thioglycolate broth. Advantage: helps in growing bacteria for biochemical testing. Used in blood culture to detect bloodstream infections. Pure culture techniques. Pure culture techniques isolate a single microbial species for study. Streak plate method. A loop is used to spread bacteria across an agar plate in a pattern. Results in isolated bacterial colonies. Advantage: provides a simple method to isolate pure colonies. Used in identifying individual bacterial species from mixed samples. Spread plate method. A diluted bacterial sample is spread evenly over an agar plate using a glass spreader. used to count viable bacterial cells. Advantage: allows accurate quantification of bacteria in a sample. Used in microbial load estimation in food and water. Pour plate method. Bacteria are mixed with molten agar and poured into a petri dish. Colonies grow both on the surface and within the agar. Advantage: useful for counting bacteria present in liquid samples. Used in enumeration of microbes in dairy and pharmaceutical samples. Tube dilution method. Serial dilution of bacteria is performed in tubes with broth. Helps in reducing bacterial concentration for obtaining isolated colonies. Advantage: useful for determining the minimum inhibitory concentration, MIC, of antibiotics. Used in drug susceptibility testing, anaerobic cultivation of bacteria. Anaerobic bacteria do not survive in oxygen and require special conditions for growth. Anaerobic culture methods. Thioglycolate broth. Contains a reducing agent that lowers oxygen levels. Anaerobes grow at the bottom, while facultative anaerobes grow throughout. Advantage: simple method for testing bacterial oxygen requirements. Used in culturing Clostridium species, e.g. C. tetani causing tetanus, gas pack anaerobic system. A sealed jar with oxygen-absorbing chemicals to create an oxygen-free environment. Advantage: allows growth of strict anaerobes. Used in diagnosing anaerobic infections like gas gangrene. Anaerobic jar. Contains a catalyst and oxygen-removing agents to create an anaerobic atmosphere. Advantage: provides an effective method for anaerobic bacterial culture. Used in growing Bacteroides and Fusobacterium species. Roll tube method. Anaerobes are cultivated in tubes with oxygen-free agar. Advantage: ensures anaerobic conditions throughout bacterial growth. Used in isolation of strict anaerobes like Clostridium species. Conclusion: the growth of microorganisms requires appropriate media, culture methods, and environmental conditions. The choice of media depends on the type of microbe and the purpose of cultivation. Anaerobic bacteria require special conditions for survival and growth, making anaerobic cultivation techniques crucial for diagnosing infections.